Dear learners, we are going to discuss about the immune systems, its different components and immunoglobulins. What do you mean by immunity? Immunity is the ability of the human body to resist almost all kinds of organisms and toxins that tends to damage the tissues and the organs. What is an immune response? It is the reaction of the cells and the fluids of the body to the presence of a substance which is not recognized as a constituent of the body itself. Means reaction to the foreign particles, pathogens, fungi, bacteria, viruses. What are the different mechanisms of immune response? There is a series of mechanisms. We start with the pre-immune mechanisms. The antigens penetrates into the tissues and adsorption of the antigen takes place in the immune tissue. Then the immune mechanism starts in where the migration of APCs in T or B zone, antigen presenting cells is APC. Okay. In the T and B zone, peripheral organs of the immune system processing of antigen. There are three stages in this. One is endocytosis of antigen. Then the stage 2 is the cleavage, and stage 3 is the expression of the degraded particles of antigen at the MTC in complex with MHC1 and MHC2. Stage 4 presenting to Th cells, helper T cells. Okay. Step number 3 is suppression of the immune response, that is autoimmune disease can run, and step number 4 is immunological memory. The first line of defense, its goal is to stop the pathogens from entering the body. The components are skin, which acts as a barrier to invasion, sweat, which has the chemical which can kill the different pathogens, tears, which has lysozyme, which has powerful digestive abilities that render antigens harmless. Saliva also performs in the similar manner that of tears. Mucus can trap pathogens which are then sneezed, coughed, washed away or destroyed by chemicals. Stomach acid destroy the pathogen. Second line of defense, if a pathogen is able to get past to the body's first line of defense, an infection starts. The body can rely on its second line of defense. What happens here? First, there is a non-specific response, that is phagocytosis, and then it is followed by an inflammatory response. Phagocytosis is carried out by the process of ingestion and then digestion by the pathogens. What do you mean by cellular immunity? It is a type of immunity when the body develops large number of lymphocytes which are specifically activated against foreign agent. These activated or sensitized lymphocytes have the ability to attach to a foreign agent and to destroy it. So, cellular immunity is composed of sensitized T lymphocytes. Cellular immunity is a process of developing phagocytic cells involved in the expulsion of pathogens from the intracellular space. Phagocytic cells in return attacks the pathogens and destroy them. On intracellular level, the macrophages are the main cell protectors which have the specific receptors, that is immunoglobulin, through which they affect on the pathogens. Its aim is to combine the phagocytosis and specific antibodies to fight the pathogenic environment. What are the different cells that are involved in cellular immunity? T lymphocytes, macrophages and natural killer cells. Macrophages present antigen via their surface MHC to T cells. The T cells recognize the antigen through their specific receptors. A specific T cell clone becomes activated and begins to proliferate. The activated Th lymphocyte becomes effector cells that can secrete the cytokines. In the antigen processing and presentation, the protein pathogen processed and converted into peptide to bind to the MHC molecules on APC, that is antigen presenting cell, to be presented on the T cell. Okay, this is the cytotoxic T cell and this is the helper T cell. This is major histocompatibility complex 1, class 1, and this is class 2. Reds are the class 1. Okay, here you can see the whole process in a diagrammatic way. The next process is the process of T cells activation. We all are going through the second line of defense. Okay, helper T cells express interleukin 2 receptors and secretes the cytokines, which includes the interleukin 2, which auto activates the helper T cells. Antigen presenting cells, that is APC, release interleukin 1, which act on both APC and helper T cell to promote their activation. All mentioned interactions lead to the activation of mature Th cells. These mature Th cells proliferate and differentiate into the effector's antigen specific Th cells releasing cytokines. Some of them become memory cells which provide secondary immune response. The cytokine released by this process from activated Th cells can activate the macrophages, natural killer cells and B cells. The T cells activation process is continued by activated CD8 TC cells which proliferates and differentiates into a clone of effector cells, that is CTLs. The effector CTLs targets and kill target cells, that is nucleated cells, which express the major histocompatibility complex 1 infected with virus, tumor cells or graft cells. Next is the process of macrophage activation. The activated helper T cells secretes interferon gamma, which activates the macrophages and increases their ability to kill ingested intracellular pathogens. The process of activation of macrophage, natural killer cell and cytotoxic T cells infiltration and proliferation of inflammatory cells stimulated by cytokines released from helper T cells is important in protective mechanism against intracellular pathogens. Cytokines stimulate other effector cells of cell mediated immunity and humoral immune response and mediate the following. They attract the monocytes, macrophages and lymphocytes to the site. They activate the macrophage to kill the intracellular microbes. They promote the activity of CD8 cells which directly kills the virus infected cells, tumor cells and graft rejection. They activate the natural killer cells, increasing their cytotoxic functions. They stimulate the B cells to differentiate into plasma cells that secretes the antibody. Sometimes the second line of defense also fails, and as a result the third line of defense comes in activations. Okay. The immune system recognizes, attacks, destroys and remembers each pathogen that enters the body. It does this by making specialized cells and antibodies. Okay. For each type of pathogen, the immune system produces cells that are specific for that particular. Humoral immunity. A humoral response is carried by antibodies which are produced by plasma cells. The plasma cells are derived from activated B cells that are produced in the bone marrow. The humoral immunity promotes the development of normal operation antibodies. The aim of bacteria and viruses is to enter the cell and destroy it. The principle of action of antibodies is the interruption of receptor linkages between the pathogen and the cell. The result is to breakdown of interaction between the pathogen and cells due to blocking it by the effector molecules or antibodies. Antibody is a protein that is produced by the B cells that is used by the immune system to identify and neutralize the foreign objects such as bacteria and viruses. These are the series of steps which are carried out: agglutination, opsonization, neutralization, activation of the complement takes place. After that, inflammation occurs, and at the end the antibody dependent cell mediated cytotoxicity is shown by the antibodies. Now we are going to discuss about the different immunoglobulins. IgG, the most common forms, and it represents the 75 to 80% of serum immunoglobulin G. It has only antibody that is capable of crossing the placenta to give passive immunity to fetus. It is important. Has longest half-life of 23 days. It activates the complement and stimulates the chemotaxis. Next is IgD. It is present on the surface along with IgM. It activates the basophils and mast cells to produce the antimicrobial factors. Next is IgE. It is bound to the surface and mast cells of basophils, and it destroys the parasitic worms and participates in allergies. Next is IgA. This is the structure. Present in the body secretion, it provides the protection against proliferation of microorganisms in this fluid. It aids in the defense against microbes and foreign molecules penetrating body via cell linings of these cavities. They provide the passive immunity to infants through mother's breast. IgM, it is a pentameric structure. It is the first immunoglobulin class that is produced in a primary response to an antigen. It is found on the surface of beta cells. The antigen receptor of beta cells has 10 antigen binding sites and more effective at stimulating complement. The Fc receptors on phagocytes binds to IgM, that is the process of opsonization. Interferons are proteins that are made and released by the host cells in response to the presence of pathogens such as virus, bacteria, parasites or tumor cells. These are also abbreviated as IFNS. They allow for communication between the cells to trigger the protective defense of the immune system. that eradicate the pathogen's tumor. Okay, there are three types: gamma interferon, alpha and beta interferon. Complement system is a system that helps the ability of antibodies and phagocytic cells to clear the pathogens from an organism. It is a part of innate immune systems, not adaptable, doesn't change during lifetime. The complement system consists of a number of small proteins which is found in the blood, and it is synthesized by the liver and normally circulating as an inactive precursor, that is proproteins. When stimulated by several triggers, proteases in the system cleave specific proteins to release the cytokines and initiate the further cleavages. Memory cells, which do not react right away but are held in reserve for later infections. The secondary response that is carried out by memory cells is different in three ways. Memory cells produce the antibodies that bind with greater affinity to the antigens that the antibody produced in the initial response. Memory B cell, the response time is much faster than the primary response, and it is an antibody producing cell. Memory T cell is an infection fighting cell. A greater number of antibodies are produced. So with this we finish this topic. Thank you.